Okay, thanks, Lala. So I'm going to talk to you today about how RNA diagnostics fits into one of the major trends in medical technology today, what we're trying to do, and some of the challenges that we're going to face in the future. So, to start off, many of you probably know that about 10 years ago there was an announcement that the first draft of the human genome had been mapped, and that this was going to lead to a revolutionary transformation of medicine. And in the middle of an election year, President Clinton stepped up to the podium with Francis Collins and Craig Ventnor, who had been the groups that had mapped the genome, and said this is going to transform medicine completely in the next 10 to 15 years. Of course, that was a lot of hyperbole, and for the last 10 years we've been riding down this the curve of disappointment, and many of the euphoria that surrounded this announcement has popped from the balloon. So now that we're at the bottom of the curve and we're looking up, and we're starting to see real transformative developments coming out of our understanding of the genome, and that's what we're trying to do at RNA Diagnostics. So we're in the personalized medicine business. So what is personalized medicine? In fact, I can't imagine anything that's less personal than being treated for something by your doctor, but personalized medicine today means the use of your genomic information about an individual, whether that's proteomic or your DNA, and particular mutations that you may carry, to tailor particular treatments, whether those are drugs or diagnostic diagnoses, for particular patients' individual makeup, so that these patients can be treated more effectively, and we can reduce the cost of delivering those care to those patients, and this is a great benefit to society as a whole. So we're starting to see some real significant advances because of the genome, and this is where medicine is going today. So the first area in which genetic knowledge has been applied to medicine has been in determining a lot of predisposition towards a developing disease, and these range from very useful understandings of whether or not you're going to get diseases like Parkinson's disease. If you have a genetic mutation for Parkinson's, you will get the disease, and maybe you should start planning for it. If you know you're going to have a predisposition to getting heart disease, you may want to change your behavior and get a little more exercise and stop eating potato chips. On the other hand, knowing that you have a predisposition to hair loss is probably not going to change your life very much, though if you found the pathway and designed a drug to solve that problem, you'll make a lot of money. So this is one area that's been useful, but the real, one of the most significant areas in personalized medicine has been the development of drugs that are focused on particular subsets of patients that are, for which these drugs work and have been very successful. Two of these drugs are in the cancer area. Have been a breast cancer drug called Herceptin, which is a $7 billion drug from Roche, and Erbitux, which is now owned by Merck, which is a $1 billion drug. So these drugs can only be prescribed for patients who have particular genetic mutations that qualify them for the drugs, because they will work for those patients and they won't work for patients who do not have these mutations. And this is important because these are very expensive drugs and they have side effects. So again, we're delivering personalized care that's effective for patients and at a lower cost and a lower burden for healthcare systems around the world. And this is the future. This contrasts with what has happened in the past, where drugs have been approved based on being effective for a low percentage of the patients for whom they have been prescribed. So you'll see in cancer chemotherapy, for instance, less than 25% of the patients who are given these drugs derive any benefit. And this is where RNA Diagnostics is focused, and I'll talk now briefly about RNA Diagnostics. And as we're having dinner tonight, I'm going to play a 5-minute video that we made of our clinical advisors talking about the product, and don't feel it necessary to sit at the table, but you can go eat and listen to them as they talk. So we started this company 2 years ago, and last year, just about this time, we were very fortunate to have won the TiQuest competition against a very diverse and sophisticated bunch of companies. It was a great help to us in getting raising our visibility and getting us going. So in the last year, we put together a clinical advisory board that's second to none around the world. We've raised about $2 million. We've advanced our product development, and now we're bringing the product into the clinic in a bunch of clinical trials that I'll talk about. So this is the problem. This is what I like to ask people. You're a breast cancer patient. You've had your first cycle of chemotherapy, and the question we like to ask is, would you like to know whether that chemotherapy is working or is not working now, or do you want to wait 26 weeks, experience all the side effects of the chemotherapy, and find out that it was pointless to take that chemotherapy because it didn't work? So this is the very important issue that we address at Rna Diagnostics. So here's the problem. 50% of breast cancer patients will receive chemotherapy as a part of their treatment, yet less than 25% of those patients will derive any long-term benefit from the chemotherapy. This means that we're vastly over-treating this population of patients with drugs that have serious side effects and don't benefit them. This is the major problem facing breast cancer specialists today. Today they have no way of knowing which of those patients they treat with chemotherapy will benefit and which of them won't benefit. So at Rna Diagnostics, we've developed the first real-time chemotherapy guidance tool to solve this problem. The RNA disruption assay, or RDA, is the first diagnostic test that predicts early during treatment how a patient will respond to chemotherapy. So after an RDA test, those responding patients can continue the chemotherapy with confidence, while the non-responders can be switched to alternate therapies. This spares them the toxic effects of the ineffective chemotherapy and also allows them to be put onto alternate therapies that improve their chances of survival. This is a very important discovery. We have validated this with a large-scale clinical trial that was conducted here in Ontario, and we protected this discovery with a group of broad patents. So the significant benefits of this technology are threefold for the patients and for their physicians and for healthcare payers around the world. Patients are now able to avoid a lot of unnecessary and toxic treatments and be put onto therapies that have the potential to improve their survival benefits quickly. Their docs now have a test that they can use to effectively manage their chemotherapy regimens, and very significantly, and as John has indicated, very importantly for healthcare payers, this is a method that they can adopt and promote to reduce the cost of delivering cancer treatments around the world. So we think this is a real win-win-win situation for the patients, their docs, and for payers. A little bit on the technology. That's an RNA molecule. An RNA is a very complex molecule. What we do is we look at the RNA of the patient's tumor after the patient has been treated with chemotherapy. And if we see a large degree of fragmentation of that molecule, it means that that patient will continue to success and that all the cancer cells will die. If we don't see an adequate level of fragmentation, that means continued cycles will not result in success, and it will not result in complete eradication of the cancer cells in the tumor site. And that's what success is measured as a pathologic complete response. This is a very significant discovery. When we tell oncologists around the world about this, they say that if what we do is true, and we know that it is, that this is going to become a standard of care in cancer chemotherapy around the world very quickly. So this is how we implement our technology in the clinic. After the patient has received her infusion of chemotherapy, her oncologist will take a fine needle aspirate biopsy of her tumor, immediately stabilize this sample in a RNA stabilization test tube, which will then be sent to a central lab where it'll be RNA will be isolated and run across a bioanalyzer instrument. It's at this point that we use our proprietary algorithms to analyze the data coming off the bioanalyzer and produce an RDA score that's then sent back to the ordering physician. It's very important to note here that we are using standard chemistries to isolate and stabilize the RNA and a standard bioanalyzer to do the analysis. Our proprietary position comes in with the algorithms that we have to analyze the data. So one, it gives you a very low capital cost to bring this product to market, and it gives us a very strong proprietary position in defending ourselves as this becomes known in the marketplace. So as I said, we have a very strong intellectual property position, and we are building that as we speak today. We have a strong protection on the use of this RNA analysis for assessment of a tumor response to chemo for all cancers, and we are developing proprietary algorithms in conjunction with the math department at Waterloo University, which again strengthen our IP position. So this is a big market. In breast cancer alone, there are over 1 million new cases in the BRIC countries in the developed world each year. We expect that we anticipate a total market opportunity of over $500 million in breast cancer, and we're forecasting sales of $100 million within 6 years because this solves such a serious problem. There's nothing like it in the marketplace, and the oncologists that we talk to say that if this really works the way we say it does, that it's going to become a standard of care and it's going to take off very quickly. Competing methods are, one, physical palpation of tumors by their docs. This does not indicate whether or not that patient will proceed to complete success, because most tumors will shrink after they've been treated with chemotherapy. So it really is not very useful. And the same with imaging. Most tumors will shrink, but only a small percentage of them will proceed to complete success. You may have heard of these gene panel tests, the Oncotype DX and MammaPrint. These are gene signature tests that analyze the gene signatures of tumors and predict whether those patients, based on that, will have a 10-year recurrence of the disease, where they have a high or low probability of that recurrence. They don't say whether those diseases, whether the chemotherapy that would then be prescribed to these patients will be successful or not. So this is what we do with RNA diagnostics. We say you've been prescribed chemotherapy because you have a strong likelihood of having disease recurrence, and you are taking the drug now. Now we tell you whether or not it's working. So we're planning to move to market first in Europe. We're going to Europe first because this type of chemotherapy is practiced more widely in Europe, and the regulatory environment there is much more friendly, and we're working with our partners in the German breast group and with the group in Italy. To the US shortly thereafter with what's called a lab developed test. This is a method of entering the US market, working with CLIA-approved labs delivering a testing service. It doesn't require FDA oversight at this point, and we can do that within 24 months. We do have an expectation that there will be further FDA approval requirements for the US, and we will be building our files to supply that information to them as we move forward. We're working with groups here in Canada, in Germany, and in the US and Italy on clinical trials, and these are all in the early stages right now, so that's going to be a big challenge for us to manage this as we move forward with them, working with distribution partners to deliver the service around the world, building our management team, raising capital. Don't be alarmed, but it takes a lot of work to build a company like this from the ground up, so we're really excited about what we're doing, but we're also realize that there's a lot of challenges ahead, and we're working hard to solve them. So this is what we're doing at Rna Diagnostics. We've got a very unique solution to what's a very serious problem. It's a big market. We should be able to expand this to other markets around the world, other markets, other cancer markets. We have strong IP, which we're building out. We've generated our first revenues from clinical trials, and we are anticipating generating more revenues to support our programs with clinical trial customers. Again, it's a low capital cost opportunity, and we're going to get to market quickly. So it's we think it's a real winner. We've got a tiger by the tail, and be happy to take any of your questions. Now, as you go to pick up your food, if that's okay, I'll play the video.